Hello and welcome everyone to Nowhere You There is another day, another episode. Today we'll be doing biology, nervous system for the MCAT prep. The MCAT covers nervous system in two different topics or themes: the biology and the behavioral science books. So today we'll be uncovering the nervous system in the biology. We'll be talking about the nervous system, reflexes and responses, and the biological side of it, really. So, we're going to be talking about how is information transmitted just in most neurons. So, the way information in a neuron is transmitted is dendrites receive signals. So, dendrites, um, if you don't know, they're basically just the ends of a neuron or that extend like that. All right, so these receive a signal, okay, and then the signal goes through a cell body. A cell body is covered by a myelin sheath, and then the axon hillock carries the signal using action potential. We'll be talking about action potential later. So part of a neuron, like I said, is the myelin sheath, which is produced by the oligodendrocytes. And so the main two jobs of the myelin sheath is maintain electrical signals and increase the speed of conduction down the axon hillock and the cell body. So the nerve terminal is flat to maximize the transmission of the signal to the next neuron. So it's flat the way the neurons are, or the terminals are, they're flat. So in the central nervous system, nerves form tracts, neural pathways, in which they carry only one type of information group, and then they carry them and send them to nuclei. So they form tracts, neural pathways, carry one type of info group, and then send them off to nuclei. Alright, we'll be talking about glial cells. What are glial cells? Glial cells have supportive and structural roles in the nervous system. So, you have two types. You have astrocytes and you have oligodendrocytes and Schwann cells. I mean, is that technically three? I would count these two as one type. So, with the astrocytes, their job is to nourish neurons, control the transmission of solutes from blood to neurons. Neurons are like any other cells. They also need nourishment. So what the astrocyte does is that it makes sure neurons get the nourishment and transmits or the control the transmission of solutes from blood to neurons. Oligodendrocytes and Schwann cells form the myelin sheath, the one we spoke about earlier, which maintains electrical signals and increases the speed of conduction. With actual action potential. Action potential's main purpose is to release neurotransmitters through the reversal of membrane potential to pass the signal onto the next neuron. So, the way neurons communicate with each other to send a signal from one neuron to another is through this, the action potential. Alright? So, we're gonna talk about the mechanism of the action potential. So, this is the first bit of information. The resting potential of a neuron is negative 70 MV. Alright, that's millivolts. When neurotransmitters bind to the receptor dendrites of another neuron, the neuron is depolarized, making membrane potential less polarized, helping it move close to zero. Alright, so the first part is basically binding of the neurotransmitter to the receptor. Alright, then second part is sodium and potassium. They have a tug of war. So with between so sodium plus 60 and potassium negative 70 MV. Alright, and then they get to a resting point, which is negative 70, like I said, the membrane potential. Alright, then the Na plus K plus the sodium, potassium ATPase pumps potassium in and sodium out. Eventually, it reaches a threshold membrane around negative 55 MV. So it needs to reach the threshold. There could be any failed initiations, but it keeps trying. So It pumps potassium, reaches the threshold, and then after the threshold, it begins depolarization. All right. So when the threshold is reached, right, at -55, a large number of sodium channels open. So now the potassium channels are closed, the sodium channels are open, and depolarization starts occurring. So a positive charge just flushes into the cell. All right. Action potential reaches its peak here. Depends from cell to cell, but let's say here is +40, a lot of positive charges, and then now it reaches peak, it needs to come down, needs to be neutral after it send the message. All right, so it opens the potassium channels to influx and repolarize. All right, so at the top now sodium is done, they close the channel, they open up the potassium channels and repolarize. All right, but there's a problem. Sometimes I have a hyperpolarization occurs. So with hyperpolarization, it means that there's too much potassium that's being influxed into the cell. But then eventually the K plus channels close and return to resting potential, which is, like we said, -70. All right. So summation is basically an additive effect of multiple signals. What is additive effect? It's combination of two or more drugs together. So basically just having two or more um, drugs that affect your neural system, and summation is basically a combination of multiple signals. So you have temporal summation, multiple signals are integrated during short period of time. Spatial summation, additive effects are numbered, and location are based on incoming signal. Alright, impulse protonation, so travel of action potential membrane, action potential down the axon to initiate neurotransmitter release. Well, of course, you need action potential, you need something to trigger the neurotransmitter release, so that's what impulse protonation is. Now, let's take an example here. Do you guys know the pufferfish? Even if you don't, search up a pufferfish. So, tetrodotoxin is the toxin found in pufferfish that blocks the sodium channels, alright, in the diaphragm, and the diaphragm can't depolarize anymore. The dangers of that means you can't breathe, you start to suffocate because there's no more action potential. There's no, like, one small piece is missing, right? And then boom, you go, you suffocate and stuff like that. Ever wondered what do they use and how it works in surgery rooms? Anesthetics, local anesthetics are being used by blocking Na plus channels and block the transmission of pain. Um, Basically, while doing surgery, they don't want you to feel any pain at all, okay? Because feeling pain wake you up most likely, will cause um, bad stuff. I'm not a doctor yet, but you get me. So increased axon length increases resistance and slower conduction. So what a local anesthetics does, it increases the axon length, and which increases resistance and slows down the conduction. Of course, it's not 100% blocked, but rather the axon length is increased. And then you have saltatory conduction, signals hopping from one node of river to another. So nodes of rivers are basically, here, let me draw it. So let's say this is a neuron. Alright, so that's a node of river. Alright, and then node of river jumps. The signal jumps. Oh, wrong one. Neural circuit, alright, we'll be talking about that next, are called reflex arcs, and they control reflex behaviors work through interneuron pain impulses by the spinal cord. So the spinal cord controls the reflex arcs. So you got two types. You got monosynaptic. What is monosynaptic? Monosynaptic starts with a sensory afferent presynaptic and then it goes and sends info to the motor efferent postsynaptic motor neuron response to it. So basically, um, when the knees stretch, it's presynaptic and sends information to tendons to contract in response to injury. So basically, it's unconsciously doing that, alright? So monosynaptic sensory efferent presynaptic, alright, and then sends information to the motor efferent postsynaptic. Next, you have polysynaptic reflex. So these are sensory neurons that fire onto the motor neurons and utilize interneurons passes signals to multiple motor neurons. So for example, you consciously control withdrawing your leg when stepping on something sharp, and then use your conscious control, like I said. Alright, so conscious, this is like autonomous, right? Automatic. Okay, well, thank you everyone. This is the summary of nervous system biology. Let me know if you have any questions, suggestions. Don't forget to leave a comment and like and subscribe, and let me know your opinions. Thank you so much.